Cells are amazing systems. Through the last decades, studies have proven that they have, like us, kind of a sense of touch. They can feel the mechanical properties of their environment. We know this because, for example, stem cells can differentiate into different types of cells just with changes in stiffness of the substrate they are on. But how can they sense? Scientists have proven that the cytoskeleton is one of the major actors. It makes sense because this is what gives shapes to the cell and transports information from membrane to nucleus. To go deeper, here is an experiment. We want to design a material with tunable stiffness without changing its chemical component. A solution is to make pillars. They can bend, and the bigger the length, the easier it is to bend them. We made a soft and a stiff one, and we deposited cells on it. We saw that on soft substrates, cells were round, whereas polarized and stiff. To better understand, we changed a little bit the composition. We tuned the quantity of crosslinkers inside the cytoskeleton. We saw a change in the comportment. If we reduce the crosslinkers, cells on soft substrates began to polarize. And inversely, when we increase the crosslinkers, cells on stiff substrates became round. Since crosslinkers increase the stiffness of the cell, we deduced that polarization was linked to the stiffness of the cell. So we derived a model, and we found that the polarization was actually a function of substrate stiffness over cell stiffness. More specifically, if stiffness of the cell was too high compared to the one of the substrates, cells would be round. Hence, knowing the stiffness of the cell, we could design material with appropriate stiffness to shape the cell round or polarized.